Imagine a computer that doesn't just run on electricity, but is, in a very real sense, alive. This isn't science fiction anymore. It is the reality behind the CL1, a groundbreaking device developed by the Australian company Cortical Labs. Unveiled to the world on March 2, 2025, the CL1 represents the first commercially available biological computer. At its core, it integrates living human brain cells with silicon hardware, creating a hybrid system that learns and processes information in a way that is fundamentally different from any machine built before. This new form of intelligence, which they call synthetic biological intelligence, promises to be more energy efficient and adaptable than traditional AI. The core idea behind the CL1 is to harness the incredible power of evolution. For billions of years, biology has perfected the art of information processing through neurons. These cells are masters of learning from their environment, adapting to new challenges with remarkable speed and using very little energy. Traditional computers, based on silicon chips, are incredibly fast at predefined calculations, but struggle with the kind of flexible, intuitive learning that a simple living organism can perform. The CL1 bridges this gap, taking the best of both worlds, the precision of silicon and the adaptive learning of biology, to create something entirely new and revolutionary for the field of computing. What makes the CL1 so special is its departure from the rigid logic gates of conventional computers. Instead of ones and zeros flowing through fixed pathways, information is processed by a dynamic living neural network that can physically change its own structure as it learns. This system doesn't need a separate external computer to function, it is a self-contained intelligence. By creating a platform where researchers can interact with and study these living networks, Cortical Labs has opened a door to understanding not just the future of computing, but also the very mechanics of thought, memory, and consciousness itself. This technology isn't just a lab experiment, it is being prepared for widespread use. With commercial availability planned for the second half of 2025, Cortical Labs is making this advanced tool accessible to researchers around the globe. They are offering the CL1 units for direct purchase, as well as through a cloud-based service called Wetware as a Service. This approach allows scientists to run experiments on the biological hardware remotely, democratizing access to a technology that could redefine industries ranging from medicine to artificial intelligence. It marks a pivotal moment where biological computing moves from theory into practical application. To understand how the CL1 works, we need to look at its four main components, which work together like the organs of a body. The first and most vital part is the biological component, the neurons. The system contains a culture of approximately 800,000 living human brain cells. Crucially, these neurons are not taken from human subjects. Instead, they are grown from what are known as induced pluripotent stem cells, or iPSCs. These are created by taking adult cells, like those from a blood or skin sample, and reprogramming them back into a stem cell-like state, from which they can then be guided to become neurons. This clever process avoids the ethical dilemmas associated with embryonic stem cells. The second component is the electronic interface, the silicon chip that acts as the bridge between the digital world and the biological one. The neurons are carefully cultured on top of a specialized chip called a multi-electrode array, or MEA. This isn't just any chip, it's a stable, transparent plate made of metal and glass, featuring 59 electrodes. These electrodes can perform two critical functions. They can send electrical signals into the neural network to provide input, and they can listen to and record the electrical activity that the neurons generate in response. This two-way communication is the foundation of the entire system's ability to interact with data. Of course, living cells cannot survive on a chip by themselves. This brings us to the third part, the life support unit. This sophisticated system is often described as a body in a box because it provides everything the neurons need to stay alive and healthy for months at a time. It's a self-contained environment that meticulously controls temperature, maintaining it at a steady 37 degrees Celsius, just like the human body. A microfluidic system continuously circulates a nutrient-rich liquid to feed the cells, and a filtration system removes waste products, while a gas mixing unit regulates pH and oxygen levels. This box is the miniature world that keeps the biological brain of the computer thriving. Finally, the fourth component is the software layer, a kind of biological intelligence operating system, or BIOS. This software is the translator and the teacher. When a user wants to give the CL1 a task, the software translates digital data into a specific pattern of electrical pulses that the electrodes deliver to the neurons. It then monitors the resulting cascade of neural firing, decodes these biological signals back into a digital output, and most importantly, provides feedback. This software implements the learning rules, guiding the network to improve its performance over time without needing any external computer to manage the process. The process of using the CL1 begins with input. Imagine you want to teach it something, like how to recognize a sound or navigate a simple maze. The software layer takes the data representing that task and encodes it into a language the neurons understand, patterns of electrical stimulation. These signals are sent through the electrodes on the chip, zapping specific areas of the neural network. This stimulation acts as the system's sensory input, much like how our ears receive sound waves or our eyes receive light. The neurons, upon receiving these signals, begin to fire in response, passing electrical messages to one another across their connections. This is where the magic of biology takes over. The network of neurons doesn't just passively transmit the signal, it processes it. The initial stimulation triggers a complex cascade of activity that ripples through the entire culture. The way this signal travels and how the network responds is not predetermined. It depends on the current state of the connections between the neurons, known as synapses. The software records this resulting storm of activity, capturing which neurons fired and when. This pattern of firing is the system's output, its answer or reaction to the input it was given. At first, this response is essentially random and meaningless. To make the system learn, it needs feedback. This is where a concept from psychology called reinforcement learning comes into play, guided by a principle known as the free energy principle. Neurons, by their nature, seek to create a predictable and stable environment for themselves. When the CL1 produces a correct output, the software rewards it with a predictable, orderly pattern of electrical stimulation. If the output is incorrect, it receives a chaotic and unpredictable signal, which is energetically unfavorable for the cells. Over time, the neurons physically adapt their connections to favor pathways that lead to the predictable reward signal. A famous real-world example of this process is when an earlier prototype of this technology, called Dish Brain, was taught to play the classic video game Pong. The system was given information about the ball's position, and its neural activity was used to control the paddle. When the network successfully made the paddle hit the ball, it received that nice, predictable electrical reward. When it missed, it got the chaotic punishment. Incredibly, within just 5 minutes of this closed-loop feedback, the neural network began to improve its performance, demonstrating a clear ability to learn a goal-directed task in real time. This showcases the system's power to learn dynamically. The potential applications for the CL1 are vast and transformative. One of the most promising areas is in drug discovery and personalized medicine. Scientists can create neural networks using cells from patients with neurological disorders like Alzheimer's or epilepsy. This allows them to test how different drugs affect these specific diseased cells in a living, functioning network, providing far more nuanced data than static cell cultures or animal models. This could dramatically speed up the development of new treatments and reduce the high failure rate of drugs in human clinical trials, ushering in an era of truly personalized neurology. In the realm of artificial intelligence, the CL1 offers a path toward more efficient and adaptable systems. Today's large AI models require enormous data centers that consume megawatts of power. In stark contrast, a full server rack of CL1 units uses only about 1,000 watts. Their ability to learn quickly from sparse data could be revolutionary for applications like robotics, where machines need to adapt to unpredictable real-world environments. Furthermore, studying these biological networks provides invaluable insights for fundamental neuroscience, helping us understand learning, memory, and the basic principles of neural computation in our own brains. However, this revolutionary technology is not without its significant challenges. A primary hurdle is longevity and scalability. Currently, the neuron cultures can be kept alive and functional for several months, after which they need to be refreshed. Scaling the system up from 800,000 neurons to the billions found in a human brain is a monumental engineering task. There is also the inherent variability of biology. Unlike identical silicon chips, each neural culture is unique, which presents challenges for standardization and reproducibility. Overcoming these technical obstacles will be critical for the technology's widespread adoption. Finally, the use of living human brain cells naturally raises profound ethical questions. As these systems become larger and more complex, discussions about the potential for consciousness or sentience, however remote, are unavoidable. Cortical Labs is proactive about this, working closely with bioethics committees to establish clear guidelines and ensure the systems remain non-sentient and focused on specific tasks. Despite these challenges, the CL1 stands as a landmark achievement. It represents a fundamental shift in our concept of a computer, blurring the line between machine and organism. This technology may one day not only power our devices, but also unlock the deepest secrets of the human mind.